two different ways of calculating the allele frequencies. And one of them is the easy method, and the other method is also called the easy method. They're both easy, all right? But one of them works with genotype counts. And so if you're given the counts, then you say, well, fine, I've got the counts. Let's go ahead and calculate the frequencies of the allele directly from the counts without going through this intermediate step of having the genotype frequencies. Or maybe somebody only told you the genotype frequencies and never told you the counts that those frequencies are based on. Well, then you want to use the other method. All right, so the first, and both methods sort of rely on you channeling your inner accountant if you have such a person in there, right? So it usually has like one of those little caps and a vest, I think is what they have, a little old-fashioned calculator on the side and pencils in the ear. So you have to find that person and bring them to the forefront because they both just require that you don't lose your wits. So in this case, the first method that uses the genotype counts, we want to figure out what is the frequency of the CR and what is the frequency of the CW allele. And it relies on the idea that each individual has two alleles. Right? Each individual has two alleles. So if you're interested in the frequency of the CR allele, you know that all these guys, the homozygous individuals for CR, have two copies of it, and these heterozygous individuals have one copy. And then it relies also on being able to realize that yes, we have 500 individuals, but we have 1,000 alleles, twice the number of individuals in the population, the number of alleles that are in the population. So our first method would say, well, let's take 2 times 320. Each of these 320 individuals has two alleles that are CR, plus 160, which is the number of alleles that are found in the heterozygous individuals, and divide that by 1,000, which is the total number of alleles in the population. So that should be 2 times 320 is 640, 740, 800, 800 divided by 1,000, so that's 0.8. Okay? That would be the frequency of the CR allele. We can call that whatever we want, but maybe we'll call it F subscript CR, right? That would be 0.8. Now you know that the two frequencies, the two allele frequencies have to sum to 1, so it's very tempting to say, well, that means that the frequency of the CW allele has to be 0.2, right? That's a true fact. But it's a true fact if we didn't screw up here, right? So we can also do a quick check and make certain that's true. So it's going to be 20 times 2, the number of alleles, or CW alleles in these homozygous individuals, that's 40, plus 160, that's 200, divided by 1,000, the total number of alleles in the population, that's 0.2. So it looks like we got that right. Okay? So those are the two frequencies used in the first method. The second method just uses these genotype frequencies. And the idea is if you want to get the CR allele frequency, you realize that the CR alleles are found in these individuals and these individuals, of course. And so we take the frequency of the homozygous individuals and add 1/2 the frequency of the heterozygous individuals. So that's 0.64 plus 0.16, which is 0.8. There's the second method. I'm not going to elaborate on that method at all. All right, so far we have not done Hardy-Weinberg at all. Right? All we've done is a bunch of calculations on this very lame flower population. But now we're doing the theory, all right? So get ready. This is the Hardy-Weinberg genotype frequencies. All right, so this is where the theory comes in. We're saying, what would be the frequencies of these genotypes, which we observed to be 0.64, 0.32, and 0.04? What would be the frequencies of those genotypes if the gametes that those individuals are making just randomly unify? Right, so if you want to stick an organism to the theory, sometimes I think about marine organisms that typically spawn. As I say, the females and males just spew their gametes out in the water column, and the gametes presumably randomly unite. Okay, that helps you think about what type of organism might actually fit this crazy model. That would be fine. But even organisms that don't fit this crazy model can be modeled with this crazy model. Okay. So the first thing we have to do, or we actually don't have to do it, but it'll make it much more recognizable to you, is instead of calling the allele frequencies F subscript C subscript superscript R, we're going to call them P, Q. Right, so now you guys are saying, now we're talking. Now I recognize this because I had that in high school. And the Hardy-Weinberg says that the frequency of the homozygous CR individual should be P squared. The frequency of the homozygous CW individual should be Q squared. And the frequency of the heterozygous individual should be 2 times P times Q. So the Hardy-Weinberg frequencies are predicted to be 0.8 times 0.8, which is 0.64. 0.2 times 0.2, which is 0.04. And 0.8 times 0.2, that's 0.16 times 2 is 0.32. These are the Hardy-Weinberg expected frequencies for those 3 genotypes. Okay. Now, what a biologist would do, so if we really had gone out into nature and collected these flowers, what the relevant comparison is this frequency, the expected frequency versus the observed frequency. Right? These are what you actually observe in nature. This is what theory predicts the frequencies would be just under the operation of random mating. Okay? And of course, by eye, it looks like they're in equilibrium to me. They're the same, right? So basically, the observed frequencies are in Hardy-Weinberg equilibrium. Now, this is something I want to tell you that is a simplification we have to make for the class, but I hope you realize that in real life things don't work like this. So if I were to give you questions on the exam with Hardy-Weinberg involving Hardy-Weinberg frequencies, and then I would ask you, are the observed frequencies of the genotypes in Hardy-Weinberg equilibrium? It's going to be an exact match or not, right? And the reason why, in real life, what a biologist would do is saying that the observed frequencies are in their expected Hardy-Weinberg frequencies, they don't have to be exactly the same. You do a statistical test to decide whether the deviations you observe are so great that you can't explain them with Hardy-Weinberg. So you have to apply statistics, right? Even if the allele frequencies, genotype frequencies were generated from random mating, just because the number of samples, the number of flowers you sampled wasn't very large, you expect stochastic variation, small errors in your numbers, right? And so in real life, this could be 0.6, this could be 0.3, this could be 0.1, and maybe you would still conclude that they're in the Hardy-Weinberg frequencies, right? It would depend on how many samples these frequencies were based on. But in the class, we have a simplified version of statistics. They either match or they don't. They either match exactly to as many decimal places as you can calculate, or they don't, right? So that's our simplification we have to live with. And it's just because I can't count on you guys having the background in statistics, we don't have the time to go over that, although it's a fascinating field, I think. All right, so that's an example of calculating Hardy-Weinberg frequencies. And the first thing you might ask yourself is, why do I have to know this? Why should I care, right? And there's the proximal explanation, which is I would be testing you on this, so that's one reason. But there should be a better reason than that, right? Because that's not a very satisfying answer. And the reason is that Hardy-Weinberg, well, I'll list out a number of reasons, but it is actually an important result because it tells you what's happening, what we expect to happen in population, what should happen in genotype frequencies and allele frequencies when nothing else interesting is happening. Sometimes you need to have what's called a null model. I'll describe that in a bit. A null model against which you test your observations. If you see deviations from what you expect, then something else is going on. So for instance, if we see deviations from Hardy-Weinberg frequencies, significant deviations, then we either don't have random mating or something is changing the allele frequencies. Right? So it's important. It's a very important result for just that. But there's also lots of practical reasons too, and I'll go through those right now. We might call this, why should I care? Okay? So the first point that people who develop the theory want to make is that variation is not lost from a population with only when you only have random mating going on and your population size is very large. Right? That's what the theory says. And remembering, going back to sort of these incorrect models that Darwin had about how inheritance works, so-called blending theory, blending type of inheritance, those types of models actually, every generation of random mating, you lose variation from the population. Okay? So with just random mating going on, Hardy-Weinberg tells you that allele frequency should not change, the variation is not lost. And we can actually prove this. So let's go ahead and do a mathematical proof, right? I'm going to call P prime, assuming that I must be getting close to the end. Okay, P prime is going to be the frequency of the allele in the next generation, and P is the frequency of the allele in the current generation. All right, so there it is. So what we want to do is calculate what is the frequency of this allele in the next generation. So we get out our mathematical hat and we say, okay, we have P prime, and we put the equal sign there, and they say, okay, what next? All right. What is Hardy-Weinberg? What do they have to say about the frequency of the allele in the next generation? Well, I'll give you a hint. We want to use the second method I described for calculating allele frequencies. We're going to be counting up the frequency of this allele in the homozygous individuals, which is P squared, right? And we're going to add to it half the frequency of the heterozygous individuals, which is 1/2 times 2pq. And then we're good to go, because now it's all algebra. So we go ahead and say, okay, that's equal to P squared plus, well, 1/2 times pq. And then we note that q is equal to 1 minus p, so we say, great, that means P squared plus p times 1 minus p. With me? This is a mathematical proof we're doing. This is exciting. So that's P squared plus p minus P squared. Which looks like this guy goes, and that guy goes. So that leaves us with p. Right? I just did a proof for you. I just proved that the allele frequencies don't change under the influence of just random mating. Pretty cool, huh? So now you should be convinced, because it's a proof that can't be wrong unless we made a mistake. So the important point I want to make here is, with just random mating and with very large population sizes, you do not lose variation from a population. That's to say, the allele frequencies don't change under the influence of just random mating. All right, so there's a kind of theoretical result you might care about. Here's something cool: human genetics. So Hardy-Weinberg is applied all the time in human genetics. And as an example, there's a so-called DNA fingerprint. All right. This is a way that, well, DNA fingerprint is used in all sorts of different contexts. So, for instance, it's used in paternity analysis, or just as a child really mind type of thing. But it's also used in the criminal justice system, right? So the idea being, that some genetic sample is left at the crime scene. Right? It could be a number of different things. It might be semen in the case of a rape case or something, but you have a genetic sample, right? And you've collared some suspect. Okay? We think this person did it. Right? Usually guys will say, we think this guy did it, right? And you note that at the crime scene, at some locus, the genetic material, this is the sample at crime scene. So you note that the genotype at this particular locus is a, little a. And then here you have the suspect. So the first thing you note is, if this individual is big A, little a, what's the probability that this individual left the sample? Well, it's 0, right? Unless there was some lab error, but it's 0. Right? So that's not so interesting. So let's take the case where the suspect has the same genotype. Now what's the probability that individual left the sample? Well, it's not 1. At least I don't know what it is, just based on without having any theory, right? In order to calculate probability, now you have to have some model or theory in order to calculate this probability. And Hardy-Weinberg provides the theory, right? So what you do is you say, well, we have to know what the probability of a randomly chosen individual from the population having this little a genotype is. That's what you need to know. And in the absence of going out and genotyping every individual in the United States or the world or whatever your population interest is, you need to have, which isn't done by the way, you need to have theory to calculate these frequencies. Hardy-Weinberg, like I said, provides the theory, and this is how it works. Every individual who goes through the criminal justice system is genotyped at 13 different loci. Okay, 13 different loci. The database that these go into is called the Combined DNA Index System. Right? The Combined DNA Index System, otherwise known as CODIS. And it's this database of frequencies of alleles from these different individuals that provides the raw data for Hardy-Weinberg. So the idea being, if you want to know what the probability of forming little a, of randomly sampling an individual who has little a is, well, you know that the frequency, you have to know what the frequency of the little a allele is. That comes from the database, which means that the frequency of this genotype from Hardy-Weinberg is FA, FA. So as an example, if the frequency of the little a allele is 0.01, then the frequency of that little a genotype in the population would be 0.01 squared, which is 1 in 10,000. So based on just, with this made-up scenario, if the frequency of the little a allele in the CODIS database were 0.01, then the probability of randomly sampling an individual from the population that has that genotype is 1 in 1,000, or 1 in 10,000. Right? And the only way we can make this calculation is by doing the Hardy-Weinberg thing. Right? So it relies on Hardy-Weinberg theory. So I don't imagine any of you will be, well, I hope you're going to be finding yourself in this situation, but now at least you know the theory that would be telling somebody, some prosecutor in a court saying, and the probability that Jane left the sample is, and they usually put out numbers like 1 in a billion. And they say, how can they get a number of 1 in a billion out of this type of information? Well, it relies on the fact that when you multiply numbers that are between 0 and 1 together, the product has to be still smaller. And so it's not just one locus that they're dealing with. But if you match all 13 loci, you're multiplying 13 numbers that are between 0 and 1 together. Some of those numbers might be quite small. So the overall probability of a match can become 1 in a million, 1 in a billion. Sometimes you can get probabilities of matches that are 1 in 10 billion, which you say, well, there's only 6 billion people on Earth, or 7 billion, I forget what it is, it changes so rapidly. But how can that be? Well, it's because the model is saying just multiply those probabilities together. Right? Now you know, right? At least if maybe you knew already, but now I'm telling you this is the basic idea of how these probabilities are calculated. You can complicate it a little bit by recognizing there's population subdivision and whatnot, but the basics are right here. So there's one reason to care, I suppose. So what time we have? Still okay. Rare diseases. Shoot. You know what? I'm going to. I need to remember them. I'm going to stop on rare diseases. We'll finish up the important Hardy-Weinberg. Then we'll get the things that are more. I don't say more interesting, but they're not Hardy-Weinberg.